We'll keep looking.
I guess we'll
This is really fascinating. I mean, this is just
keep the
such
moves,
interesting morphology, but
either keep the
you know,
moves
to be
shorter
perfectly
or
honest, I don't know what we're
the
looking
zoom
at.
shorter.
Well, I'm with you there.
Straight down in the center, going to the left right now.
Center to the left.
Right.
Something of interest.
No, we gotta keep moving right now.
Fish?
Yeah, just
I don't see
Watch it.
fish.
Sorry, one sec. We're just coming up to a cliff. I just want to see what it is to make sure there's not a safety concern.
Below the screen right now.
just I would just move on. You're behind him.
Yeah,
There's plenty of fish in the sea.
it's very true.
Pilot, do you by
I
any
heard
chance
that.
see a fish down there?
Yeah, it's science. We saw the fish.
Okay, good. We're gonna
I do
keep
not
moving.
right now. If anyone sees one, they can shout out though.
Okay.
If you look at the
Thanks, pilot.
quad split on top right, you actually see Sirius is pointing straight down at the ROV. So he's sort of zipping.
Video, if you just
ahead.
said something, it was very quiet that I could not make out.
No, I wasn't. I was talking to the science.
Oh, sorry.
I don't know why we were not stopping to look at that fish.
Now you're no longer behind me, but you are underneath me.
It's improvement.
Another small fish coming lasers.
Yeah, we're on about average 30 degrees
Okay, I
slope.
see a video.
So if we call in a move,
I will move towards it.
We just snap on it.
I think what we did there is we called in a move and then the
yeah,
ROV
give us
just
that,
stopped
go
and
for it.
zoomed for the entire move.
Now
That'd be
there's
nice.
a fish. That looks like a reef fish to me.
What is that, Bruce?
And we could just keep moving since
Yeah,
we're
that's
still
good.
behind.
Yeah.
Oh,
Yeah,
that's a
red fish.
Is that another one of those
You
sea
got a little bit of
stars?
time right here.
The red thing?
no. That's a that's a
Bumped it, but
No, it's a fish.
yeah, looks like maybe a scorpionid. I don't know.
Yeah, a fish.
White.
A red fish, right?
Redfish.
I'm going to zoom your sonar.
So, Shirley,
I think they're good with that one.
Chris Moss from the Smithsonian here. That sea star you were looking at,
I don't know.
Pterodactyl, probably Hymenaster. It's a deep sea slime star.
Snap on the red and then we just
Did you say a slime like SLIME?
yes, quick snap on
Yeah.
red as I come up.
Most pterasterids produce a copious amount of mucus as a stress or defensive
just keep
response.
cruising. We just,
When you collect them on submersibles and you bring them up, the boxes are usually filled
It's like
with
a nice
threads
fish.
and threads of mucus.
Ugh.
That one looks like one of many species that occur throughout the deep sea. They're extremely difficult to figure out because
let you keep
they
going, get
are
ahead.
cosmopolitans.
Yeah.
They live in almost every ocean.
Yeah,
But
just come on out. so I
they
can see
often
what I
have very few characters because the bodies are completely gelatinous or partly gelatinous. The skeletons are very reduced.
We're good
and,
positioned, and as far as moving,
there
we're
are
good too. So
a
Yeah,
couple of other
seriously
genera that
stationary,
are
right?
similar
Yes.
to them
Okay.
that are very difficult to tell apart.
Copy.
Special
I think the ridge is
in fact,
winding around, so I guess just try to
examination
find
of
the
that
high
specimen
point
would
and then I'll move
probably
Sirius back to you.
could conceivably
Yeah.
give me a different
If you
identification.
look at high pack, you're
It
way
looks
off.
pretty much close to
Oh, yeah.
I mean, not that
to the
not
slime
the high pack's
stars or
correct.
to the Hymenaster we often see at this depth. So
That's very cool. I've never seen anything like that.
the first one we've seen so far.
Yeah, yeah.
Although looking at Sirius sonar, it is roughly aimed towards the bulk of the ridge.
160,
Yeah.
165.
See a stalked crinoid.
Washley's interest in this?
No, we'll just keep going.
Copy that.
Yeah, we've had pictures of those guys. Not on this dive, you're right.
Okay, we're getting a good distance out there again. So as soon as we find that position,
Yeah, I think there's a flat part right above
Squat
me.
lobster over to the left.
It's a little.
Yeah,
I'm seeing
It's a little
our
confusing
participants in the
on-
chat
so I
log
guess maybe
happen to
Sirius
notice
should
all of
go-
this coral
You wanna aim
debris, so
straight
you can
towards them and
see
we'll try to
These
move
little
you back
white,
to the ridge.
those like
Yeah.
white
finger-like
mounds.
things.
Do that
Those
like
white
20
mounds
meters
are
that way
just
or something?
branches.
I am turning towards you, pilot.
All right.
So
just
dead
gonna do
branches
a-
of
above
coral
lasers.
I
skeletons.
think that's going to be
So,
prime.
So
above
they got to
lasers.
be uphill somewhere.
I'm looking at,
Let's see what's
well,
above.
I'm looking
Up
about,
the cliff.
20 due south.
Snap.
yes.
All right. We'll do that. And then we can move along the ridge after.
Sorry, video. Say again. I lost you in the voices.
Immediately where the lasers are. we can
So if you
snap
folks
on.
want to take a
yeah,
look
go
at the
for it.
Sirius image, the
Okay.
vehicle is pointed toward a big crack, and it looks as if there may be a huge boulder that is sort of partly separated
Oh, I see that.
from the wall of the
All right.
ridge that we're traveling along. So this might be something that just kind of slid down a little bit from further up.
Can we get a ship move? 20 meters, bearing 180 degrees at 0.2 knots.
So you're moving.
So right now
to
the
south,
ROV is
Rodney?
pretty far ahead of Sirius,
Yes, sir.
All right.
and so we're going to move the ship
You're going to
in the
basically
direction
get us
up-
right back to
up
that ridgeline that you're
the
on
track
right now,
of the
'cause you can
dive.
see it
Yes.
all the way.
Yeah.
and give the ROV a bit more
Sirius
slack.
is just off to the side. So.
It looks like it is winding.
Can we look at that crinoid?
So,
Looks
yeah.
like there's another crinoid hanging on
yeah,
the,
yeah.
the steep
Tether.
part of
Right
that
above
block.
lasers.
Stalked crinoid. Yep.
Yeah.
and an
video
urchin
for
right on the
quick sensor.
edge.
Try to rise to meet it.
Just keep going, I guess.
Okay. Yeah. Just keep rising.
Thank you. One out. Go, pilot. That
Pilot,
screen
do you
you're
think
looking
that
at,
it might
is
be
that,
possible there were some rocks,
the temperature?
loose rocks underneath
Yes.
this little
Yeah, it
ledge?
just skyrocketed.
Or are we not
comparatively.
in a position to be able to do anything
20%.
like that?
Let me
change
go
the
back
going
and see real
out so
quick.
that it includes that.
This
But
is
that's
the
that warm current
yeah.
coming off of the.
this is the
the
only
bottom
place
there.
I've seen
It's
anything
shooting
that
up,
looks
that's why
like
it's
it
shooting
might
up.
be loose and could be picked up.
Yeah, let me just reposition and take a look.
And just to
Thank you
note,
very much.
pilot, that you do have a good warm current again.
Yeah, I just
not too
felt
far
it.
from you. Okay.
Yeah.
I could
picked
see it
up pretty
spiking.
good.
Yeah,
it
there's a
spiked
pretty
a couple
strong
degrees.
current for
Yeah.
the pilots coming
20%.
up the wall
Yeah,
of
watch
this
these. We
feature.
just felt a pretty good current right there, so I just need to take a moment.
But if it were possible, perhaps to settle down on the ledge below the crinoid, there are some rocks right under the crinoid.
Just change the quad split upper right-hand quadrant to be the co-pilot's screen. You can see that he's monitoring the temperature change. And we came up over this ledge, you can see the increase in the temperature that he's recorded.
Do you see anything that looks
Yeah,
loose?
that was a big spike.
We're looking for a sample.
Rock sample?
Yeah.
Keep my eyes up. Oh, that looks in situ.
Yeah, it's really likely that the rocks that are sitting down on the lower ledge have fallen off this upper block as it broke free from the cliff face.
Yeah, I'm getting a lot of current right here. Yeah, wash leads. Do you see loose rocks?
Well, I'm thinking, excuse me, I'm thinking that just underneath, boy, my voice is getting all
No,
crackly
I was
here.
thinking.
Pardon me, folks at home. I have a bit of a cold. I think you can see arms of a squat lobster sticking out on the right. And maybe some of that material is loose. I'm not
down
real
there?
sure. They look kind of
No,
big.
up to immediately right of lasers.
Oh, okay.
The one that's about
Oh,
50 centimeters wide.
yeah, I don't think those would be possible samples.
Yeah, they're pretty big.
They're large, and this is a bad spot for current right here.
Okay. Well, we can try later.
Yeah, that's just not a good spot right there. But yeah, I don't think those would be too large anyway, so.
Yeah, I think you're right.
Okay, pilot, starting to get over you.
Thank you for the effort, though.
Oh, no problem. We'll find you a rock. Don't worry.
Time to. Oh, that current's really blowing you back, huh?
Yeah, look at that.
Yeah. Yeah, you're still in it. It shows the
Wow.
temperature gauge.
serious image. Look at
What's
that.
the temperature up to?
11.7.
Isn't that amazing?
Yeah, look at
I mean, this the whole
I
big
know.
thing just cracked off
know.
the side of the
I'll pick up over you, but definitely move
wall
forward,
here and slid down.
because I don't think we're done with our move yet either.
Copy that.
momentarily changed stream 3 to be a full screen
Okay,
of
I
the
got you good delta.
co-pilot's
The ship just stopped, so
screen.
Sears should be
took
stopping in a
another
second.
additional spike.
When we get a chance, I wonder if we could look at these yellow polyps
How's the
down
current
here.
doing, Pat?
The yellow
I
polyps?
think they're scleractinian, and I think
Want to look at the
where a
yellow
lot of the coral
polyps?
debris is,
Yes.
that these
His
are
current-
called
yeah, he got into it a
Down
second
in the
ago
lower
on
right, I
the
guess.
updates. We had a
and
strong
that would
warm
be a
current
new
coming up.
we haven't documented so far here in the
I think I'm
Marianas.
just kind of protected.
Okay,
on this
Chris.
wall.
Actually,
Yeah, I
it's
see what
still
you
going
mean.
up, dude.
Is it?
Yeah, we
Yeah,
have a
there are like
temperature now
4 or
of
5 of them
13
down
almost.
here. Thank you, pilot.
So there's something up ahead.
Sorry, Washley, you do want to look at the yellow polyps, right? Is that correct? Or
That's correct.
okay. Yeah, just give me 1 second to set up.
Okay, what am I looking for here? Diva is saying these look like cup corals.
Did we get our Titan
Now,
back?
now there is a name I can pronounce.
Actually, kind of looks like it, huh? All right. Video, if you want to come in.
And Scott France is telling us that these coral colonies look like some of the debris that we saw down below collecting in the crevices. You can see,
Try to rotate
you see a fragment
slightly.
just to the left of the base of this coral.
Is it moving?
Now, you coral folks,
It's
I'm
0-1,
not
yes?
absolutely certain this
Washley,
is
say
the
that
same
again. Sorry.
species, Aguchisomnia serpentina, which we have in Hawaii, but they're characterized by forming these sort of
Pilot
really
repeat.
long, tube-like skeletons, you know, where at the very end of it you've got a single polyp coming out like this. And I think a lot of the debris in the cracks are a result of these
Trying
things.
to give you a good visual here or something.
Oh, it's beautiful.
so these guys don't form a colony per se. They just form, I mean, they're
Right,
just a
getting
single,
old. Yeah.
single animal,
Current in the
solitary
current as well.
animal.
Yeah, you can see the current.
Yeah, and you can look in the inside of one of the tubes there on the upper right,
Yeah.
where the animals actually died. And so I guess they just keep elongating their skeleton. They just keep growing, and they're just sitting on the top of it, keep growing out like that, and they form this elongate
Hey,
skeleton.
Nav, where should I normally position
Thanks,
that
Chris.
Titan for him?
Washley, are we good at the polyps?
I guess on the, I usually leave it
Roger
on the
that.
shilling
All right, we're gonna lift off.
or the craft. I don't usually look at it very much.
I'm very impressed with the serious view.
Manipulating it all.
I bet, you know, it looks like as we go up there, there are going to be lots more of these things. Oh, yeah. Look, look at this. Her voice can't
I
squeak
know.
because she's got a cold, but wow. I'd be squealing if I could, folks. Look at all these guys. These guys look like they're all dead, except for the yellow ones.
The stock crinoid that's walking, and we can stop there for a second.
Yeah, dead center.
Yep.
Yeah.
Oh, so this is one that's lying on the bottom.
Coming
Yep.
back up.
So we have a Patty substitute
try
that
to
just
set real quick.
came in while
Okay,
Patty
you can
goes
float
down and
past
has
it if
some
you want.
lunch.
Slow past. All right.
Yeah.
And
Nobody
who's
cares about
who's joining us?
Hi everyone, it's Casey. I'm here to keep Shirley company while Patty's at lunch.
Keep the conversation going.
Copy that. Just gonna float forward and off.
So Shirley, have you talked about some of the crinoids that we've been seeing and how for a long time we thought that they were extinct?
Yeah, these again another living fossil. Another living fossil. Just like yesterday we saw a slit shell, and Asako said that
Bottom right.
living fossil. This is another situation where these feather stars or sea lilies are really ancient
I'll have to
animals.
sit down for that. Sorry, video.
So you can see some of this debris now too, the coral debris.
Some type of weird
It's just
ship.
dead skeletons.
So are we up on top of this ridge now?
We're still
Or are
going
we
out.
still climbing?
Yeah, we're still climbing. Yeah, we're getting to the top. Looks like it slopes off soon, but yeah.
Right.
Video. I'm gonna kind
So
of
all of
going
these
up.
corals, you can see now all these dead branches, and so leads us to think that all of these dead branches that we saw kind of in those crevices, the debris before, coming from this type of coral. Wow. What caused that?
So Shirley, this is kind of similar to about this time yesterday when we were climbing our ridge.
Yeah,
We
just gonna
saw
try to avoid
this
getting
sort of
close to that
massive
edge, 'cause
dead
it's like
area
apparently
with a
that's
few
the-
small living parts of corals, but a large chunk of dead corals.
Yeah.
Like I had all the
What
good
would have
corals.
caused that?
I don't know.
Just kind of have crinoids
Chris,
now.
do you have any ideas? Or Diva?
Tina says
Payback for the
just
shark.
some of them just naturally died.
Sent the shark in.
What would have? What's the reason we're seeing so many
I
of
think that
these
pom-pom took 'em
dead
out.
skeletons?
We're about
Yeah,
we're
could
about
very well be, and that's what.
130 meters from
Exactly.
the waypoint.
Right. That's
Maybe
what.
this
That's
is a
what.
fairly
totally
old
flattens out on top of a
bed,
ridge.
maybe,
Okay.
and they just
That's
keep.
a really sharp
You know,
cliff there, man.
there's only a proportion of these
Yeah,
that are
so I
alive
think
at any
this will be
given
over by
time,
then.
and then they just die in
It is.
place, and some of them
Top of
roll
that's at
downhill a little
360,
bit.
so only
But
about 50 meters right
this
below
looks
it,
like
100 away.
pretty much the type
I don't
of
know if
beds
we'll
of
make it there or what.
Gucci serpentina that we see in Hawaii, where you've got a lot of dead tubes, and then scattered amongst them are some live ones.
Yep. In fact, our dive supervisor Jim Newman said this about. Well, it's just a natural assemblage. There's some that are dead, some that are alive. It could be a generational thing, as you said.
Right on that ridge up ahead, we have a...
Exactly, I totally agree.
Go ahead, Chris. Sorry.
Video.
I think it's really interesting to point out now that, I mean, I think we started the dive around 500 and something, correct me if I'm wrong, meters depth, and now we're about 100 meters vertically higher. And the zonation, we can see clear zonation patterns. You know, we started off in this sort of diverse area with lots of different corals, quite large corals, and now we're in this totally different area that has these big stalked crinoids trying to get as much food as possible, as well as a couple of echinoderms still hanging around, and then these wonderful little Iguchisamnia cup coral thingies. Yeah.
Right. So we're seeing a completely
zonation patterns.
Right, so a
Going
different
to,
assemblage
turn
of
to poly 'cause it looks like that
animals
ridge
that
goes
are
up that
living
way
on
too,
the
pilot,
ledge,
if you're
living on
okay
the
with
edge
that.
of the ledge at this depth than we
And,
saw earlier.
Look at all those
Yeah,
crinoids.
So we're almost ready for another move. Once you aim at him and your sonar looks good, we could move you another 20 that way, or even 30 if the pilot wants to go quicker.
Asako says that she counted 17 in one shot.
You guys ready for that then?
Yes.
Yeah.
I'm looking 205.
Well, about 200.
200. All right.
Okay, we'll go any higher than that.
Yeah, sorry.
Lights are going away.
Currents picking me up a little. Come back down.
For those of you just joining us from home, this is NOAA ship Okeanos Explorer. We are currently using ROV Deep Discover and Sirius to explore the seafloor just outside of Mog. We are at a depth of 405 meters, and we've got just about another probably 2.5 hours or so before we start our first midwater transect of the expedition.
Good copy, Bridge.
And Casey, you want to tell us something about the Okeanos Explorer?
Sure. So the Okeanos Explorer is America's ship for ocean exploration. It's the only federal vessel dedicated to systematically exploring the large portions of our ocean that are completely unknown. So we from NOAA's Office of Ocean Exploration and Research, with a number of our partners, work to sort of systematically take a look at some of the areas of the world that we don't know much about, that we don't have those basic questions answered about what exists, how does what exists down there get there, and we spend quite a bit of time on the Okeanos Explorer looking at places that no one's ever taken the time or been able to look at. So today we're very far outside of the sort of closest city, and tomorrow we'll be going into the Marianas Trench.
Yay!
And then later today on this dive, we're going to be doing some midwater work. And the life in the water column is some of the most unexplored regions of the ocean. It is the world's largest biome and really one of the most unknown and poorly understood areas. So we've begun to spend quite a bit of time and resources invested in characterizing that and trying to answer those fundamental questions that we just don't have answers for yet.
Tell us something about the crew.
Okay, so the Okeanos Explorer has 46 personnel that sail on board at any one point in time. We have, right now, we have 21 mission personnel, but we normally have 20 mission personnel. And then we have 6 commissioned officers
just continue
as part of the NOAA Corps.
Yeah, ship is moving.
And we have...
Roger that.
On this cruise, we'll have 20 wage mariners that are participating as well. And this is our deck crew, our stewards, our electronics technicians, our survey tech. All of them are wage mariners.
To right.
Orange below the crinoid.
Did you talk about the size?
No, I have not.
I know, we're looking at our cheat sheet notes now.
So the Okeanos Explorer is just over 200 feet. I believe we're at 224 or somewhere in that range.
Like that, yeah.
And we have I guess 3 main decks and then the bridge where we have our mess deck, our forward lounge, and the library. Sort of mission spaces are up on the next floor up. And then we have the ROV launch in the back of the ship on the fantail. And we have an ROV workshop and hangar. And then we have the control room, which is where we are right now. And the dry lab, and the wet lab, which is where we spend sample processing time.
Stabilize myself.
Lasers are clear.
So we collect up to 4 samples, usually 2 rocks and 2 biological samples, and then when they come back up on the deck, then we do what we call processing, so fixing them in certain ways, and I'll go into more detail about that when we're actually collecting something, when we've identified something we'd like to collect.
Have you guys taken zooms on fishes yet?
Yeah, a couple.
Okay.
As we could. Well, they weren't darting about.
They do tend to do that. I'm not entirely sure why.
And they're kind of positioning themselves right now. Okay, here we go.
I only got a...
Okay, we're seeing
you got about
scorpionid fish here.
Rotate.
At 405 meters.
How are you?
Hold him for a few seconds. Continue to go in that
direction.
Pilot,
since there's no delay here, once the ship moves called in, you pretty much have to start going.
So does anyone on the line know a lot about these fish? Can tell us. They seem to always be sitting on the bottom, kind of
in a crevice.
In a little crevice, hiding. Is that typical? Or have we kind of startled them into their crevices?
Time to go.
All right, video, come on out, please. And I am going to push ahead.
Pilot, if you note the angle of how much we see off in the distance, we can sort of tilt the camera up so we can keep that vantage point there. It works best. It gives us much wider view of the area.
Okay, you're getting behind me and that current's catching you just a little bit.
Casey, it's Deb. I don't think we're ignoring you. I think nobody's on the line.
Looking good right now.
It's okay, Deb. I didn't think anyone was ignoring me. I know that the majority of the people on the line today are benthic ecologists and geologists, so we may not have our fish experts, but...
And it's Father's Day.
And it's Father's Day, so I hope that a lot of people are out enjoying the day with their fathers. We had an ice cream social here on the ship to make sure that we celebrated our Father's Day here.
Ton of debris.
Yeah, look at this big pile of debris.
Oh, wow.
And then just to look like maybe 1 or 2 live corals. Yeah, lots of crinoids, lots of stalk crinoids, but lots of debris.
Looks like that's going to be your direction.
How are the currents on this ridge so far?
Currents are pretty strong right here. I'm actually having trouble keeping a straight line. As soon as I lift up off this wall, it just kind of takes me. So.
Good
Well,
to
you're
know.
doing a great job.
Thank
See
you.
some, potential basket stars in the top right.
Yeah.
Oh,
Oh,
it's
yeah.
right there in front of you too.
Yeah.
There's another one of the scorpion fish.
Video. Any
So right in the
Up to
center.
the
What
left.
is that?
The
in
wash
the-
leads?
in the
Oh,
hole
there
to
too.
the
To the
left?
left, yeah.
No, we already
Two of
imaged
them.
it.
Oh, you have it? Washleads, did you say something?
Negative.
Okay, sorry. I'm going to keep moving.
Is that the same fish on the left? there?
Yeah.
Okay. Oh, you got someone coming underneath you right now, big fish.
Big fish.
Just swim underneath you.
How big?
Now he's to your
Oh,
port.
yeah.
See him on my screen?
Yeah, I see him.
Starboard
Oh, he is
side.
big.
Starboard, yeah. He's
Oh, he's
too far away.
Yeah,
he's behind me.
He gone.
Where is it?
Over
off
to
the
the
Oh, I
you
see him
it?
now.
Yeah, he's gone.
So in Sirius view, you guys on shore should have it in a couple seconds. We just saw quite a large fish.
That's the really neat thing about this setup,
I hope
because
this ridge
we can
starts wrapping
look
to your
down
port,
on
otherwise we're
Deep
gonna end up
Discover
in
and
a
see
really weird
both
place
the
and head back.
substrate, the bottom
it
around
does.
it, as well
It
as
looks
things
like it
that are
does.
swimming around it too. In this
It looks
particular
like my high
situation,
might be slightly to
the
my port as well.
view from Sirius
What
showed
size
a
depth
rather
does Hypex
large
Starfish
fish.
ahead.
I'm going to turn with you.
What's really neat is the way that the vehicles
yeah.
are positioned. Sirius
Line up my
provides additional
course.
perspective as well as
Video, you said
lighting
a
to
white
D2.
starfish.
So a lot of times as we're going along, Sirius
Oh,
will
all the way
catch
up there,
things that
towards the
D2
very
doesn't
top?
see because
Yeah.
D2 is so
Yeah.
focused on getting the really close detailed imagery. And
even wanna
Sirius gets this
tell
really
you what
great
happens.
wide view. And sometimes animals sort of are curious as to what we are, and they'll come through, but not
460.
close enough to D2 that we can actually get good
Look
enough
at that
imagery of it.
starfish
So
in there.
Sirius gets some really great imagery.
Yeah, video.
Yep, so we're
pointed that
seeing
out.
basket
one,
star at about
I'm
the
at
2 o'clock
well, I
position,
was at 180 and it
and
looked
Another
a
like
sea
sea
good.
star, white sea star.
Yeah, I think
Yeah,
it's
Washleads,
pulling you a
we're
little.
going towards
could
this
just-
white sea star too.
You could just snap on it. You can float past it.
Washleads, do you just want a quick zoom on this, or would you just want a
Yeah, quick zoom is fine.
quick zoom? All right. Yeah, video if you want to do a quick zoom.
Spot lobster too.
Move over it.
So, co-pilot, what if you center up? It looks like you're headed that way. Let's center up kind of the
Sure.
Oh
Corals
gosh, that little knobby
ahead of
thing
D2.
to
I'm thinking that's about 180.
to the right of the
okay.
sea
I
star
like that.
is a
It keeps,
little
keeps turning
lithisted
me, though,
sponge. So
right
I'm
now, but
guessing
it
that
might be
many of
far
these
away.
little
Can
white
I give them
knobs
in
are
each other?
small
Let's
lithisted
go down a
sponges.
little.
Can I say you can see 3 or 4 just in view
Yeah.
there.
I'll call
Yeah.
it in. Should we do 30?
Yes.
Get you caught up and then he'll
So my
move
colleague
a
at
bit?
Harbor Branch,
Yeah, 30 at,
Dr. Amy Wright, is a
180.
marine natural
180,
products
yeah,
chemist,
that'll
and
be
for her
fine.
postdoctoral research many years ago, she worked on these little sponges, and the idea is to be able to extract enough of the chemistry from it to do our evaluations. And these are
Rich enough.
so tiny that we never had enough biomass. So she would call it postdoc killer. So every time I
You
see
have a
these
ship
little
move.
these
20
little knobby
meters,
sponges, I
bearing
think,
180
Amy's postdoc
degrees
killer.
at 0.2 knots.
I'm going to be yanking you here for a second.
A lot of times there are just too many questions that need answers.
I know.
That's quite all right.
Good copy, Rich.
Nice thing about this
It
type
changes
of
to 20
exploration that we
because there's a
do
lot
is
of
that
corals up
the
there.
data are
Yeah.
public
Copy
data.
that.
So
Good catch.
anyone who's interested can request to use some of the samples that we collect. The data are all publicly available, so you can go back and re-examine the videos. Basically any of the data that we're collecting. And so that makes it really nice because these are data that we're collecting on behalf of the whole scientific community, and there are many different disciplines of science interested in this. So the geologists and the biologists, within each of those categories, there are numerous other sub-disciplines that and for which these data will be really valuable.
So I was actually just talking to our onboard data manager, and all of the data from this morning has already begun to hit shore. So it's for our scientists that are participating, they have access to our shoreside FTP server, and they can start going through the data that we're collecting right away,
The ship and
which is
Sirius are
fantastic.
on their way.
And then, as
Copy that,
Shirley
thank
said,
you.
all of our data is publicly available, and it's not just that it's
Might be
publicly
flattening a
available and
little.
that people have access to
Or
it, but
you
it's
just
within
moving away
60 to
from the
90
edge
days
instead
of collection,
Just moving away from
which
the
is
edge.
incredibly
knife
quick
edge now,
for
this is
science.
flat.
Yeah, this is a lot
We
different from
definitely
the
progress.
way most of us are
With
used to
divers
operating on
current.
expeditions.
With that
That's
chrono.
controlled by a chief scientist or principal investigator who has gotten funding to do that particular mission, and he or she
Oh, no,
basically
it's
has
port to
control
starboard.
of the samples
Yeah.
for a couple of
Let's
years
see.
before
Right
they
turn,
they
too.
become
so.
the data and the samples become
it was
publicly
straight from
available.
starboard before.
So this is a lot different. I mean, these are your taxpayers
So when I was going
or
to
taxpayers
Slow,
out there, and here on the ship
like a
are
nice
making it
slow.
possible for us to explore these really amazing areas. And
What are we at now?
these are part of a
13,
national monument. They're part of U.S. exclusive economic zone.
9.4.
And for our resource managers to be able to do their job, they need to know what's out here. And that's why for the last couple of days in this dive as well, we're really focusing on some of the commercial resources, the precious corals and the bottom fisheries.
Just dropped out again.
Oh.
Because there's just been kind of anecdotal evidence that they occur out, that the precious corals and the commercial fish species, benthic species, occur out here. So what we're trying to do is document their occurrence.
so it's like-
Definitely changed direction.
Yeah.
Port.
So Les Wassling
Let's see something real
is
quick.
pointing out that we're starting to see more of these
Dead branches.
Dead branches.
Yeah.
Les, are you at the UAGCC?
Yeah, the current's strong.
Hey, Shirley and Patty, it's the UAGCC, and we were just wondering how much longer we have left in the dive until we get off the bottom to do the midwater transit.
A little bit less than 3 hours.
It's still pretty steep, though.
So we're going to begin the midwater transect at 2:30
You could
2:30.
swipe them right off.
Tomorrow
just maybe
standard time.
tomorrow Standard Time. Yeah,
Little
sorry,
flatter.
2:30 in the dive. For you
So
guys,
about
that'll-
3 hours from now.
About 3 hours.
Yeah, about 3 hours.
So I'm just going to push ahead.
So we're about halfway through the benthic part of our dive.
Yeah.
Let me see. Let me find out.
Gravity.
Watch lead nav.
Go ahead, Nav.
Yeah,
Sorry,
can you
go
give-
ahead, watch lead. This is Nav.
I did that yesterday. And the day
70
before.
feet up,
Could you give us the
50
range to
down.
our next waypoint, please?
Yes, we're getting closer. We are 85 meters, and the ship is moving.
How many meters?
8-5.
Thank you. Okay.
Yeah, so nav, I'm like 70 up
Yeah.
and barely getting up.
Yeah, it's fun, isn't it?
It is fun.
Yeah, it's similar yesterday. It's hard to even know where it's coming from or if you're going up or down. Just kind of bump along and try to stay centered and serious here.
Yeah.
And you would, if you suddenly got swept away, I have the note here that you should try to turn to port to take your positive wrap out.
Okay.
You're gonna
So
spin off
guys,
into space.
I'm just throwing this out, but because there is some interest in getting
It is flattening
some
out a bit
scleractinian
more on the
samples
top of the
here,
ridge, so if you do
and
get
because
lost, you'll just
this
go
is
off into
clearly a dominant
globs.
species in this particular part
yeah,
of
kinda
the
so.
ridge,
Yep, there's,
I'm just wondering whether it might be worth
having trouble
trying to collect
turning that way.
some of this
current.
Yeah.
this
One sec video.
Gucci-somnia here.
To fight a
Maybe
lot
getting
right
at
here.
least one of the live polyps or more on a clump. What does everybody think about that?
Have you guys done any sampling yet?
Nope, we're good.
None today?
Nope, we can do that. Anyone else want to weigh in on that?
All right, video, if you want to do a quick zoom on this blob.
What do you think?
Oh, that's so
20
cool.
meters?
Oh my
At
gosh, that's so
180?
cool.
Yeah,
Whoa.
that would be
I even
good.
know
I'm
what
having
that
trouble
is.
saying it, 180
What is
though.
it?
It is
Oh,
a
I see,
demo sponge called Spongoceroides siliquaria. Spongoceroides
Yeah, it's
siliquaria
trying.
is the name of the
talking
species
about
that
the
we have
32?
in the Caribbean and Atlantic.
It is
So this is a
trying,
demo
not
sponge,
there
but all
It is
those
trying.
So
curly
already
Q
adjusted
things
to
coming
reset.
out of it
Oh,
are
okay.
vermeted gastropods. So this is very, very cool. Definitely a
Thrusters?
new
Serious
range
thrusters.
extension for this.
Okay,
We've only
serious.
seen this in the
Thrusters
Atlantic. To my best of my
overturned.
knowledge, it's
Oh.
not been
Do it
reported from the
disable and
Pacific
re-enable.
at all.
Okay, up at the top here.
Yep.
Shirley, could you punch that into the chat room, please?
I will.
Oh,
Copy that video.
that was my first Fault.
Do we want to get? Yes.
Is this something we can name after me, or is it taken?
Who asked about something being named after them? It's very cool. It's very cool. Yeah, we're gonna stay. Yeah, we're gonna.
Okay.
Yeah, pilot. What we've had a request from the shore team for a collection of one or more of the solitary these corals, the yellow corals, because it seems to be one of the dominant species in this area.
Yeah. What about the one
We're
right
thinking
front?
that you sometimes see clumps of maybe 3 polyps
Right,
together,
okay.
and maybe.
So the
there's a small
request
like
is
that.
actually if we can see one where there are clumps where there's more than one.
Okay. Yeah.
But I'm totally psyched about this Spongosaurides.
I think I'm running a little late, but the white sea star
degrees at
is
0.2 knots.
called Paranapanthia.
Yeah,
And,
I'll fly around. I
I
think I
that's a
see
new
one right
record
here.
for the Hawaiian.
180
They're
degrees.
usually found closer to the central
Yeah, sorry.
Pacific.
Now,
It's
can
really
you actually
interesting.
hold
Never
off on
seen one
that?
alive
We want to
before.
sample.
Didn't know it was
Sorry.
white.
You're pretty far ahead of me right now.
Yeah, I was
In
going to
this
move
kind of
it.
substrate,
It's just
so,
1.5 meters.
Okay, yeah, that's
Great
right.
that we saw one. I hope we'll see another one. Thank you.
Chris, can you spell that so we can punch it into the chat
So
room?
facing
Sure.
into the current should get you
Para,
hitting the most stable.
P-A-R-A,
Yes, I'm going to try to turn a
Panthea,
little. It's almost
N-E-P-A-N-T-H-I-A.
down on me right now. Well, it's pretty
Wow.
down,
That
I
may be
think.
tricky.
Thanks, Chris.
You said
That's
it was
fascinating. So
4
two
before, but
two range
of course also
extensions
straight
in
into
the course of about
rock is
5
also
minutes.
the most stable.
So what we were saying before about how unknown and how little we understand about our ocean is just a perfect point right now.
Oh, it looks like there's.
Made to order.
Oh, it looks like there are lots of the Spongosaurides too.
Better.
That
something you could
that
grab.
sponge actually is
Yeah,
very
sorry, I'm
interesting from
just
a
having a lot of trouble
pharmaceutical
right now.
standpoint
Give me one
because it
second.
produces a compound
tilt your
that
HD
has
down so you can see your
anti-inflammatory activity. So, for example, when cosmetics are formulated,
I would keep going, and you won't
sometimes people who
accidentally
use
sit down
those
too far.
cosmetics are allergic to them.
Delicate
and so,
skin.
Delicate skin, and so
Just
some
like
of these
us.
anti-inflammatory kind of natural products get incorporated into the formulas. And in fact,
Scoundrel.
the Estee Lauder Resilience line of cosmetics incorporates a natural product, a natural anti-inflammatory compound that is produced by an octocoral.
Interesting.
Yeah. And this one is also quite interesting, and so you don't necessarily need. In fact, you don't need the
Ooh, there's a good
sponge
clump.
at all anymore to get
yeah,
the
watch it.
compound.
That's-
It can
I'm
be
trying to
chemically
center that.
synthesized.
I just have to fight a lot of current.
Yeah, thanks, pilot. You
Almost
found a good
there.
clump.
I think that clump is a little bit wedged into a crevice.
Boy, look at all the dead stalks over on the left.
Yeah, so we've been seeing quite a bit of that.
I see videos point there. Do they share a base? Like if you were to grab something, you want to try?
So I don't know that we're going to get to be able to tear Shirley away from all these sponges.
I know.
Do you want me to bring you a sandwich?
No, I'm going to go down in a minute.
Let me just do a quick zoom video.
Gotta keep Shirley's energy up.
Yeah, video quick zoom.
Not
Let's
that it's
see.
very difficult.
Just so psyched seeing that Spongosaurides, I got to tell you. So psyched. Very psyched.
Oh, there's some young ones on there too.
Yeah, there are some babies.
Yeah, that's really nice. That's really nice.
My 90.
And that little knobby white thing over to the right is probably a lithisted sponge. You can see
Science, how
the
brittle
kind of the
will
5
it
o'clock
be?
position,
Alright,
some
come on, video.
encrusting demo sponges
I have a
as well.
question from our pilot. How brittle these are, Shirley?
Now, what do you think? Go for this? Or look for
Actually,
that?
I mean,
Get your right
I think
toe
you'll be
and
able to
see it's
pick them up
oscillating.
with a, you know, if you get to
Turn
go
your
to
head
the
a little
base
more.
of them.
Oh, you have auto head on. Yeah, if you can turn your head a little more forward.
The gripper has
I mean,
a
auto
blade
would do it
as
too.
well as
Yeah, I
some
don't think
rubber,
Yeah, both touching. I think would be a lot. Yeah. See,
a rubber
lot more
bumper
stable.
on the inside to maintain a good friction on whatever they pick up. But
So that was actually custom built for this vehicle by our pilots and our
you can kinda
engineers.
see
They
it.
custom
Yeah.
designed that.
that's
Pretty
that's
much all
cool.
forward, and
I've
it's
been told by a
pushing me
lot
away.
of the folks working on the ROV that a lot of the capability of the ROV
So, yeah,
really
that's
is
where I'm at
designed by
right now.
them.
Yes.
And over the years, it's improved every year by suggestions that come in from science,
So
but
then I can
also
put some
by things
joy
they
lock.
just think
That's
about.
what
Yeah, sitting over
I'd be
the
putting into joy lock.
Oh, you
the
just saw
galley
it.
table
Yeah.
and chatting.
Yeah,
It's a
try,
very bright team of engineers.
They are.
I see. It's pushing it. So even if you get it back in there and push a little harder, it doesn't stay.
Or I'm not in joy lock right now. I'm pushing full forward.
Oh.
And I'm still kind of...
And it still won't let your toe down, huh? Try, maybe move your auto head.
So if we were going for one of these clumps, I wouldn't worry about getting half of it or anything. There's a gazillion of these things all over the place. If they could try to grab it as close as possible to the base of the clump, that might be the best approach, because they could be fairly brittle.
Yep, Chris, thank you. I'll relay that. Pilot, just making sure that you heard Chris on shore suggesting that trying to grab these at the base and not worrying about taking a piece from further up the column, but trying to get the entire base and the polyp. That'd be just fine.
Base and polyp? Okay. Thank you.
You know what the other challenge is going to be?
It's up against a...
You can't open your bucks.
Oh, that's true. So I'd have to... I don't know about that. I don't know if I should... To lift up in this current. Washington, 1 sec. We just need to discuss on how we're going to do this.
I'm following you over just to stay.
I'm just going to go into joy lock briefly.
Yeah.
And as we talk, we can see if I slip or not.
So yeah, autos definitely won't work.
Autos will definitely not work.
Yeah, you can't open the box. Maybe if this is a prevalent enough animal, maybe we can look around for one that's on the very top of something flat.
Yeah. Washades, are you there?
Yes, we are. We're listening in.
Okay, so our problem is, so we're in an area of high current.
Right.
And we're on a cliff, so we can't open the bio boxes.
Right, I understand.
So we would have to lift off into the current, and then that would be
That would give you
tougher.
room to open the bio boxes,
It would
slide
give
it
us
forward.
room to open the bio boxes, but we'd be going into the current, so it would kind of sweep the vehicle away and possibly the sample.
I see.
Before we could get it into the bio box.
So do you want to try to find one of these specimens up on the top of the ridge?
That's
so you're
what
out.
yeah, if we could find one where I
Okay.
can, on a flatter surface,
Okay.
that would make things a lot better.
Yeah, I understand. Sure.
Okay.
That's good. Good
yeah,
approach.
If that's fine with you, we're just going to keep going up and hopefully find a nice flat spot.
That's fine.
These seem to have been fairly common, so it shouldn't be a problem.
Excellent. Thank you.
So for those of you on the telecon, we have
Yeah, I
our
think you might
pilots
wanna
are
move
going
me closer to 160
relocate. There's a bit of a high
degrees
current
next time
right
then.
now, so we're going to try and collect one of these corals a little bit farther upslope, potentially in a little not on a cliff face,
That's a good call.
so that they'll be able to actually open the
Thrusters are
bio
at
box
90%
and we
and
won't
you
lose the
weren't even-
sample.
You were barely able to hold it.
Yeah.
We've noticed quite a bit of high current when we've been working in the areas close to the islands recently.
160.
And in particular, it's difficult
Seems to
here
line up
because we're
closer.
working at
Look at
the
your path
edge
there where the current's
of a
bringing you.
very
Yes,
steep
wow.
scarp and
yellow
one that
blob,
is
yellow
quite
Serious, just
high.
did a
So
20-meter circle.
that presents a
Sorry, video. I need to try to get to a better spot here.
that presents a blockage to the currents, and they just sweep
Stuff
up that
seems to be
cliff,
everywhere.
just like
Yeah.
winds coming across the plains,
All right,
hitting the
so,
mountains, and going
Cirrus
upward.
is Moving. Well, Cirrus just moved in a
So
circle.
for that slime
Really?
star that we
Yeah,
saw
look at that.
earlier,
Oh,
Dave
wow.
Burdick
Yeah, it
is
wasn't
noting in the
connected
chat
before, it
room
was just a
that
perfect circle.
unless it's been a recent discovery since in the
we
last
can
10 years
move
or so,
Cirrus closer now. You
that slime
think more
star
like
that we saw before was
160?
likely a new
Possibly
Marianas
even
record as well.
150.
So three
Yeah, look
new
at the
records in the
terrain
last
here,
hour. That's
Yeah.
awesome.
That's what I'm
That's
trying to turn
incredible.
towards. It's
There's
just
there's so
a hard
much
time
living
turning there.
down here.
Yeah,
It's
we could
just
do a 150,
phenomenal.
20 meters, get you closer.
Yeah, that'd be
Yeah.
that would help a lot, I think.
Totally fine with that. I'm actually just going to kind of go drift port and see if I can get out of the current at all.
And here we're seeing more of those sponges that Shirley was really excited about here too.
Bridge nav.
Yeah, you're pulling me.
Am I pulling it? All right, I'll
Bridge,
stop.
do you copy? Oh. Hey,
Oh
can
my
we get a
goodness.
ship move 20
There's so
meters,
many of the,
bearing 150 degrees at
the stalks of
0.2
these
knots.
things around here. This is all of the white stuff that we're seeing. Is those are the stalks of the yellow coral? Is that correct?
Yeah, that's what we think, based on the discussions we were having earlier. And we were going back and forth between whether this is a sort of natural progression, since we're seeing some are dead and some are alive. But at the same time, this looks like a lot more dead than we had a little bit farther down the slope.
You don't seem to be on a knife slope exactly now.
No, I think it's on the to my starboard.
Yeah, you can see it on mine.
The
Yeah,
edge.
I
Oh, yeah.
went away from it because it was really taking me away.
Sure. So if we do find one of those corals on a flat area where we can open it
Oh
up.
my, yeah, you're right.
Good news is they are everywhere.
Lots and lots of dead ones.
Yeah.
Should flatten out. We're actually getting very close to the top now.
Perfect.
There's another one of the sponges in the center screen.
Head back, we're actually only
Yep.
D2 is only 30 meters away.
Straight ahead video.
Yeah, there was something with arms.
If you had to make just a visual
Yeah, let me just
estimation of
or
the percentage of the seafloor that's covered by
snap it.
material
Oh, yeah, go
from
for it,
these
Snap
these little corals that have the single polyp, I would guess that you had 15%
That's alive or dead?
that are just
right.
just the surface covered by the stalks of things. This must have been a really prolific area for the growth of these animals.
At some point, I'm sure it was.
Now, what
See,
are
it's rotating.
the dark animals?
We believe those are
should.
crinoids.
Should?
Yeah.
Oh, right. Okay.
Fluffy thing in the
It's a little hard to tell still.
The dark fluffy thing.
Is that a crinoid?
I think so. Yep.
different
Looks like.
for today.
Yeah, let me just try to keep steady. One second video.
It looks
All
like,
right. Yeah, if you want to snap zoom.
looks like you're in your direction there.
Yep, so that's a crinoid, but those are a little different than the stalked crinoids that we were just seeing before.
So these are attached to the seafloor without a stalk.
Copy that.
Right.
They have just little gripper,
just little gripper legs. Yep.
gripper-like things.
This is a really weird crinoid. Two-tone colors and tons of arms.
Yeah, I agree. It's beautiful.
Chris, it also looks like there's some of those, the arms are a little bit smaller than the others.
Been chewed off by somebody?
Pull for full specimen.
No idea. Boy, that's got a lot of arms.
Tilting up.
Sure does.
Good.
Could it be two?
Lasers are on.
Copilot, are lasers on?
No lasers
I don't
on.
think
they
so.
are
I
now.
think it's
It
all
should be.
one individual.
Yeah,
Oh, wow.
You just have to come out for lasers video.
Double check though.
Okay, just a
Yeah,
few
they're
more
on.
seconds.
Yep. On new video.
It's a chromatulid crinoid.
And
Thank you,
Ooh,
Tina. I
a
had
lovely name.
I had forgotten the name of it.
Let's move boys back home.
When I first started working with OER, I wanted to see a crinoid so badly because for a long time people didn't know that they still existed. And the very first dive that I did with Okeanos, I was so excited when we saw our first crinoid. I was like jumping up and down.
There he goes.
I'd never
great.
seen one.
He went
Oh,
right
that's
underneath you
great.
again.
I'd only worked in
I
shallow
see it.
waters
Is that
where
it right
we didn't
there?
have them, so it was really exciting.
Yeah.
Oh,
Got the pilot camera.
There's a
on.
fish.
shot off.
There
and
Oh, we
is
got
a
the pilot
fish.
camera.
I
down
think
the
he's
slope.
stuck. I'm just going to sit
No,
still.
he,
I can see the
he swam
tail
off this
of
side.
a fish.
he's stuck.
Pilot, look at the rail cam.
There, we have a
We have a fish right in front of the ROV, so close that you can't see it,
Stuck.
except in one of the small cameras. It looks like it's
Oh man.
oh,
trying to
Just left. You almost got him.
No, wait,
feed
come back.
on something on
the
the
ROV.
Hey, we're not edible. Go away.
Yeah, he left.
He's gone.
Oh.
Man.
He's gone.
Copilot, I don't want to say you were slow.
I think he went off with something, though. I think there was
You're a
something in
little
his
quicker.
mouth.
We might have got him.
He took one of the
I was
cameras.
looking at the other fish.
Uh-huh. Yeah, I guess we should have said port rail
Just kidding.
instead of that one.
We'll be more descriptive next time.
So it is flat.
So there's
out
another one of
here.
those
If we did
crinoids.
see a clump
Actually, a
of
couple of
yellow
them, more of them
flat
are in view
spot,
now.
we could-
Yeah, maybe.
Is that move done too?
Pilot.
Should be.
Yes.
Move
It,
is done.
it might be.
Light of lasers
It might
or
be
something.
nice to move me to the
Another one of those
port.
So would
Just to
anyone
get,
like to
where
sort
he
of
is, 'cause he's
describe
been kinda staying away from the
what
knife
we've
edge.
been seeing
Video,
over the
sorry, I
last
keep repeat. I heard
probably
light of lasers.
10,
Above
20,
lasers.
30 meters or
Above
so as we
above.
transit up slope?
You're trying to get out of the current.
I was
Oh,
trying-
there's one of those
Yeah.
snails.
Okay.
Oh, I
yeah.
see it. The snail?
Yeah.
Oh, okay, yeah.
Let's take a look at that guy. This looks very much like the one that we saw previously.
Split
So
shell.
Diva Moan is telling us that this is another slit shell, so similar to the one that we saw yesterday.
Right.
So we've probably
It sure looks
never
like
seen
it.
these on this
It's
ship.
beautiful shell.
Seen them last 3
And the
days.
animal inhabiting it is the original occupant rather than a hermit crab. We saw a tiny one yesterday sitting in a hermit crab sitting in the shell, sitting in the bowl of a sponge.
Man, he's rushing off too.
It always amazes me. Every time we get really close and zoom in, you see
Oh,
so
so
much
much.
more life.
Absolutely.
Every inch is covered with some sort of life.
It looks like it, doesn't it?
I mean,
You just wonder about the
Try to really take
biomass of
it,
the
yeah.
oh
oceans.
yeah.
Just kinda, there we go.
So this is Deb, and for some of us geologists, we'd be really happy if there wasn't quite so much life on some of these rocks. It'd be very helpful if we could actually see what's beneath all that goo.
Little too much bioclutter for you, Deb?
She called the BioLife.
Oh yeah.
Goo.
I was just having a conversation with one of the
Sorry,
other
video. Rocking a
geologists,
little.
and we were talking about the fact that they ruin all these great geology shots with biology.
Good.
That's true. You know, it's like the two of us, Deb, we could be driving along the Pennsylvania
I don't think it's
Turnpike
the
and we're going, yeah, oh, forget the trees. Wow, look at that anticline. Look at this. Oh, there's a fault. And that's because giant bulldozers have ripped the ground apart to make the road, and here we are ogling the inside of the earth.
So in our frame of view before,
Hopefully we're not driving.
All right, let's see.
in our frame of view there, we just had 3 separate sponge
I
species.
think if you find a perfect flat place where you can really
So,
get some
Deb, I
toes
just
in,
want to
then
point
go.
out that if
Otherwise,
this
that would
is
take a long
carbonate,
time.
it's
Yeah,
the
there's a lot of
biology that made this rock.
'Cause there'll be more up ahead. Anyway.
All right, all
Yeah,
right.
You have a point, although
Yeah, we'll
it
just,
does seem that
look for
most
a
of the
perfect spot.
geologists, of course,
Let me
all of the
turn
people
more
who have
towards
weighed
you
in
and
are
towards
hard
the
rock
slope there
geologists.
again.
They're all
Pilot are pretty good at that.
volcanologists. Do you
Yes,
think
maybe
this is more
not.
likely to be
Okay.
volcanoclastic? I'm still waiting for the carbonate geology person to get back to me.
Side and back over again.
We'll go slow, and I will let you know video.
So inside of the
Okay, looks like we're
caldera,
ready
there
for
are
another
quite a
move
few
if we
corals,
throw out
so
there. Should we do,
it's possible
30
that this
meters
whole area
time?
was corals at
yes,
one point.
but I think we might want to do a 140 because that looks like to be
Alright.
the location
that.
for me.
Well, actually, inside of the caldera, in the very center, there is a volcanic
Ready for
dome,
that, pilot?
a plug
Yes,
of
absolutely.
viscous lava that has oozed out, just like the
Rich
one that you would see
Yeah,
in Mount
you can
St.
see the
Helens.
tether.
If you took
Yeah,
a look
that
at the
ship
live
moves
Mount St.
per
Helens
report.
Oh yeah, it's
140
site
degrees
online,
at
and
0.2
right next
knots.
to the
Alright,
dome,
video,
there's
I
a
don't wanna
little
spend
cinder
too much
cone.
time out here.
Just want to make sure it's been a while since.
So not
Yeah,
everything in there is
still a lot
covered
of
with
light.
coral. Well, everything is covered with coral.
So we have more
Oh,
stocked
So the car's kind
crinoids
of in
here
your face
and
now,
some
right?
more of those
Swinging
basket
the tether
stars.
straight up.
yes.
It's in
And the
G2
yellow
space,
encrustation
which
is a sponge, is that right?
southwest.
Is that what we thought, or is that a coral?
I can't actually see it well enough.
Can you? Oh, it's right behind the pilot's head, coming off on the right-hand side.
I can't see it. It's a little small.
Okay.
I see these little off yellow or sort of off white yellow, light yellow blobs there.
Right.
Those are the sponges that Shirley was really excited about.
No, there was a patch of bright canary
Oh, wow.
yellow stuff.
Look at the over on the
Sirius
Sirius
view.
Getting as
view.
much tether as I can in this terrible current.
So
Yeah,
in Sirius
that's
right now, we're seeing quite a few
I'm probably a little bit low, but I mean,
There
I'm
are
safe
basket
everywhere.
basket
stars
stars likely hanging over.
living on the
That is what
steep
matters.
edge of this ridge.
Snap on the thing that's immediately inside the laser.
They seem to like to cluster right along the
White sponge.
edge of whatever
170.
promontory there is. They like to be right up at the top of the ledge, but off into the water. As the currents are sweeping up that side of the ridge and bringing all their food to them.
Temperature's up to 13.7.
All right, video, if you want to do a quick snap.
Detritus delivered.
Detritus delivered to your doorstep.
I don't know what that is.
Some type of a sponge.
Here's a sponge.
Describe and they get a still image of it. That is sufficient for still image. So we can move on.
All right, on new video. All right. Oh, we got some more tether now.
Yeah,
nice.
You got some play now.
Let's keep going up on a flat spot.
So just off in Cirrus View, we just had a siphonophore
Oh, there's a big fish again.
drift by.
It's stuck in us by chance?
No, he's
And what is a
swam over
siphonophore?
you. Oh, there he goes. From the Titan.
Thank you for
Sirius.
telling me.
It is a gelatinous, or another fish too, dudes. Another one again through the Sirius View.
The fish are swimming around the backside of the ROV. They're smart.
Coming for me. I don't feel safe.
They don't want to show up on Facebook.
If you can't see me with your nice fancy cameras, you can't have me.
That's right.
Oh wow! Look at all the stock crinoids again.
Yep, top blade.
And the basket stars too, all on the edge.
And there is an urchin. And you still haven't told me what a siphonophore is.
1 second, sorry.
Snap away video.
Nice.
So they are
nice little guy. Look at this. Let's see the tube feet.
So siphonophores are a gelatinous colonial organism that are quite common in the water column. And later today when we do the water column transects, you'll probably see some absolutely gorgeous ones.
A lot of them have bioluminescence, right?
Yes, a lot of them do. And they're so pretty. And they make these odd structures that it's kind of hard to describe, but they're usually sort of barrel-shaped and in a long line.
Oh,
they're really complicated and beautiful.
Okay, we're clear with that.
All right, come on out.
Yeah, we were able to see the tube feed on this urchin as it moves along.
Spot to get that specimen, so we did not forget. But we still see them everywhere, so we will definitely sample it.
And I still don't see anything that looks like a loose collectible rock. Very frustrating.
What is the rock here likely?
That's what we're wondering about. That's why we want to grab it. I think actually these are, we're going over a surface that's composed of volcanic clastics. Some of the dark rocks embedded in this material may be pieces of the underlying lavas that got ripped up during eruptions that produced this ridge. But then seems to be a lot of ash cover over it. And Maug did blow its top once, and that's why there are 3 little islets left. It had a tremendously explosive eruption sometime in its history that cleared out its throat entirely, blew off the top of the volcano and scattered it over the seafloor around the base.
So with all these sea stars that we're seeing, Diva, are these the same ones that you were seeing at the Sea Star City? The Basket Star City?
Looks like we're getting up close.
Or similar looking?
You're good.
I mean, possibly. We can't really tell from hair, I'm guessing. But either really closely related or the same species, I'm guessing. But displaying very similar behavior, arms extended, trying to get particles and copepods and other small animals out of the water column to feed.
So we are stopped, right?
Yeah.
Deb, I'm not sure. Do you remember what depth we saw the last aggregations at?
Absolutely not. I'm sorry.
Is that a common behavior?
Yeah, so Casey, that's how they feed. And in areas like this where there's lots of currents whipping by, that's sort of perfect for them, trying to get as much food as possible in a similar way to in which the corals orient themselves on the cliff edge and other suspension fishes do.
So guys, we are holding here for a minute while our pilots switch out to go get something to eat.